Recording is Raja sir, we can start.
Yeah. So, good evening, everybody. So, thank you for joining on this invited lecture series by the North Zone Indian Society of Nephrology. For the first talk, we have Dr. Muthukumar from New York, and this session would be moderated by Professor Dipankar Bhowmik from All India Institute of Medical Science, New Delhi, and Dr. Madhu from New York. So, Professor Bhowmik and Dr. Madhu, all yours, please.
Yeah, thank you, Raja. I think it's a great initiative on your part, so that North Zone is organizing one nice webinar, and the topic is very important. That is the DSA renal transplant, a very important topic and of great clinical value. So it's a real pleasure to have Dr. Muthukumar, who we have heard a lot, and it's nice meeting you, at least online. So the floor is yours. I think you can start your talk. Dr. Madhu, you can say a few introductory words regarding his current position.
Good evening to all from New York. It's actually a very warm, sunny spring morning here. If you see Dr. Muthukumar's first slide, it really looks like a live camp from New York today with its bright appearance of New York City. I want to really congratulate the new office bearers of North Zone Indian Society of Nephrology for taking this initiative and energizing the society by starting very dynamic academic programs. I'm really grateful to be part of this program today. And I'm even more grateful that you call my knowledgeable and a wonderful friend from all the way from Chandigarh, now in New York as well, Dr. Thangamani Muthukumar, with whom every conversation gives me a lot of pearls of knowledge and wisdom, which I always look forward to, like all of you today. And I am so happy to be here and happy to be listening to Dr. Muthukumar along with you.
Thank you, Madhu. Thank you, Dr. Bhowmik. Good evening, everyone. Thank you, Dr. Raja Ramachandran, for providing me this opportunity. It's really an honor to be here today. The topic that I'm going to discuss today is testing for donor-specific antibodies in kidney transplantation. I hope my slide is visible.
Yes. Nice.
Okay, good.
And you're audible, very clear.
Okay. So let's start with the question, what is the cross-match test? Now, cross-match test is a test to assess the immunological risk of a recipient with a potential donor. The first recorded cross-match was not for kidney transplant, but for blood transfusion as early as 1908 in New York City. The first successful kidney transplant, we are all aware, was done in 1954. But here, cross-match was not an issue because it was done between identical twins. Paul Terasaki in 1964 proposed for the first time in a patient that pre-existing antibodies could have been the cause for immediate graft dysfunction. And in 1969, this famous New England Journal paper was published, where you can see that patients who had antibodies against random donors, when they underwent cross-match where recipient and donor risk assessment was done, patients who had positive cross-match, 80% of them had, or the other way around, patients who lost the graft, 80% of them had a positive cross-match. So this test became a gold standard since 1969. And after that, all kidney transplants, cross-match has become mandatory to make sure that there is no immediate graft dysfunction. So when we say immediate graft dysfunction and we are doing a cross-match, the next question is, what are the major antibodies that are relevant in transplantation? The major antibodies that are relevant are antibodies against ABO blood group antigen and antibodies against human leukocyte antigen, which are called as alloantigens. Now, what about blood group antigen? We are talking about, we are transplanting only the kidney, not the blood. It's not a blood transfusion. But then why are we talking about ABO blood group antigen? It's because of the fact that though these are called blood group antigens, the antigens of the ABO system are present in every cell in the body, especially more in the endothelial cell. And we all have antibodies against the blood group that we do not express. So these isoantibodies can cause immediate graft dysfunction if ABO incompatibility is there. RH antigen or the RH system is not present on endothelial cells, so RH typing is not needed. Now, antibodies against human leukocyte antigen are called alloantigen, and this is the one that makes us as us. If I am Muthukumar, I am recognized as Muthukumar by my own antigen system, HLA antigen system, and if any HLA that does not belong to me, my body perceives that as foreign. These are called allo because antigens in the same species are known as allo. Let's say that we cross the barrier, and when we go for xenotransplants, we all have natural antibodies against animal antigens, and those are known as xenoantibodies. So allo antigen or allo antibodies are the antibodies that are present in the same species, but that differentiate one individual from another individual. So what are these alloantigens that we are dealing with? The alloantigens are HLA, or human leukocyte antigen. These are cell surface glycoproteins. Interestingly, these are the most or the highly polymorphic antigens that are known. There are more than 20,000 HLA class 1 allele expressed on all nucleated cells. There are more than 8,000 class 2 alleles expressed on professional antigen presenting cells. By professional antigen presenting cells, we generally talk about three major types of cells: B cells, dendritic cells, and macrophages. And in certain situations, class 2 antigens are also expressed on endothelial cells when endothelial cells are acting as antigen presenting cells. There are three major class 1 genes, HLA-A, B, and C. There are three major class 2 genes, DP, DQ, DR. Now, HLA class 2 is a little more complicated than class 1. In DP, we have an alpha chain and a beta chain. In DQ, we have an alpha chain and a beta chain. In DR, we have an alpha chain and 4 beta chains that are encoded by DRB1, B3, B4, B5. To make things more complicated, in DR, though there is 1 alpha chain and 4 beta chains, there are only 3 possible for an individual. A maximum of 2 from DRB1 can be associated with a maximum of 2 of DRB3, B4, and B5. So this is the alloantigens, the class 1 and the class 2 antigens that we are dealing with. So is HLA nomenclature that complicated? Unfortunately, yes. Now this cartoon here shows the current naming convention for HLA. You will see that HLA-A, there is an asterisk. In this example, I have provided there is a 02:101:01:02 followed by N. The A there is the locus, which is the gene name, and the numbers 02, 101, 01, and 02 are different fields. There are 4 fields: field 1, field 2, field 3, and field 4. At the end, you have several alphabets. Here, what is shown in this example is N, and that's a suffix there. Now, this field 1 is this allele group or an antigen, and field 2 is specific HLA protein. Let's not go into field 3 and field 4 today. For now, let's restrict to field 1 and field 2. Now, field 1, when we do a conventional serological typing, in olden days, HLA is like blood group antigens, HLA is determined by serology. So when we do serological typing, we write that as HLA-A2. By DNA typing, this naming convention comes with an asterisk. So you have DNA typing HLA-A 02 asterisk and a 02. Field 2 are different proteins, but that do not react with each other in serological test. For example, if we say A2101 and A2102, they are different proteins, but do not react with each other in serological tests. So currently in US, HLA typing needs to be done by DNA typing methodology. There are 3 levels at which the HLA typing can be done: a low resolution, high resolution, and an allelic level resolution. Low resolutions are serological equivalents. In other words, if it is restricted to field 1, if my laboratory simply reports the HLA typing as HLA A02 by DNA, that's called low resolution typing. When it reports to a level of field 2, say I report a patient's HLA as HLA A02:101, then that's called a high resolution typing. And if the all 4 fields are reported, then that's called an allelic resolution typing. Okay, now which part of the HLA is seen by an antibody? So let's say I have an antibody against HLA. HLA is a big structure. So which part of the HLA is seen by the antibody? Now on the left, you will see a cartoon of an immunoglobulin molecule. We all know that immunoglobulin is typically represented as a Y-shaped structure where we have a light chain and a heavy chain and a stalk region, which is known as the FC portion, and the 2 branches of the Y, which we call the FAB portion. Are you able to see my pointer when I move the mouse?
Yes.
Yes, we can see it.
Okay. So if this is the immunoglobulin or the antibody structure, this is a simple cartoon, but at this tip where the antigen binds, you have a finger-like structure. And this is known as complementarity determining region or CDR loops. There are 6 CDR loops, 3 light and 3 heavy, L1, L2, L3, and H1, H2, and H3. So when an antigen binds to this finger-like structure, it's as if holding the antigen in both our hands. So this is the configuration or the 3-dimensional configuration when an antigen is binding to an antibody. So going back, if this is an immunoglobulin, it can be an antibody against anything. So this is a simple cartoon provided as a Y-shaped structure, but when an antigen binds, it takes the form of our hands holding on to the antigen. So these 6 regions are recognizing the antigen. Now let's go to the HLA molecule. On the right side, this is your HLA class 1 molecule that has an alpha-1, alpha-2, and an alpha-3 together, and an unconnected beta-2 microglobulin chain. And HLA-2 class 2 molecule has an alpha-1, alpha-2, and a beta-1, beta-2. This is the peptide that binds. So more or less, there is structural similarity between this class 1, class 2 molecule and the immunoglobulin structure. Now, on the right, you have a cartoon, which is a 3-dimensional model where the blue is the beta-2 microglobulin. This pink is the HLA molecule, and this green is the peptide. So this L1, L2, L3, H1, H2, H3, these are the portions of the antibody that are, portions of the antigen that are seen by the antibody. So this portion is known as epitope. So epitope is part of the antigen that's recognized by the antibody. Remember, when we say HLA, it's a huge structure. When we say anti-HLA antibodies or antibodies are formed against HLA, it is not forming against the entire portion of the HLA, but a specific portion of the HLA known as epitope. So the part of the antigen recognized by the antibody. Historically, it was appreciated that antibodies developed after exposure to a single allo-HLA reacted with certain other allo-HLAs as well. These cross-reactive groups share epitopes. What does it mean? So to make our lives more complicated, though we name HLA as HLA A2, A4, A6 like that, epitopes can be shared across different HLAs, and these are called as cross-reactive groups. Now, within an epitope, let's say this is an epitope, look at this H3. This H3 portion is an applet here. What is an applet? A center cluster of about 2 to 5 amino acids that determine the binding specificity of an antibody. So at the end of the day, HLA may be a huge structure. You can form an antibody against HLA, but what determines one particular antibody from another particular antibody is just 2 to 5 amino acids. So this 2 to 5 amino acids that is in the center of the epitope is known as applet, and these determine the binding specificity of an antibody. If a donor and recipient HLA share an epitope, then that epitope will not be recognized as foreign and therefore will not provoke an antibody response. In other words, at a serological level, I may have an antibody against HLA-A2. But if that HLA-A2 or whatever the HLA that we are dealing with has multiple applets, and if the applets are matching, then these antibodies are not reacting against that applet, and these patients can be transplanted. So these days, there are certain programs that try to match at an applet level rather than at the entire HLA level. And in future, probably there will be more programs that start routine mapping of applets, and there will be applet level matching. Okay, so now, so far we learned about what are the alloantibodies that, what is the alloantigen that we are dealing with. When we say there is an antibody against HLA, what portion of the antigen is being recognized? Now, let's move on to what are the different cross-match tests that's routinely being used. At the first level that we are all familiar with is the CDC cross-match, which tests the functional potential of complement fixing antibodies in recipient circulation, which can immediately bind to and react against the donor antigen. In other words, if I am getting a kidney transplant and somebody gives me a kidney, antibodies in my serum are reacted against donor lymphocytes. So this is the output of the test is cell death, the death of the donor lymphocytes. Now, this is in a way a surrogate for what is going to happen if a kidney is transplanted in me. So I have preformed antibodies and that are going to attack this kidney. So instead of, I cannot simply test my serum to the kidney that I'm going to get in a test tube. So as a surrogate, we get lymphocytes from the donor instead of the kidney, and my serum is being reacted against the lymphocytes. So this is a functional potential of complement fixing antibodies. At the next level is the flow cytometry cross-match, which is not a functional test, but identifies antibodies directed at cell surface antigens. Now, whereas CDC cross-match or complement-dependent cellular cytotoxicity cross-match uses a functional readout of cell lysis, in the flow cytometry cross-match, only the binding of HLA-specific antibodies is detected, regardless of their complement fixing or pathogenic potential. Now, there is, in addition to CDC and flow cross-match, which are known as physical cross-matches, there is something known as virtual cross-match. Virtual cross-match is an assessment of immunological compatibility based on patient's alloantibody profile compared with donor's histocompatibility antigen. So the virtual cross-match uses the results of two independently done physical laboratory tests and does not involve mixing of serum and cells. So in physical cross-match, both CDC and flow cytometry, you need my serum and the donor lymphocytes. Whereas virtual cross-match, these are done independently and does not involve mixing of serum and cells. Instead, immune compatibility is assessed by analyzing the results of donor HLA typing that was done separately and patient antibodies against HLA that is done separately. Okay, so I just told that HLA antibodies are bad, and these are the alloantibodies that are creating problems. So why do some individuals develop anti-HLA antibodies? Now, I just told before that we all have naturally occurring blood group antibodies to the blood group antigens that we do not express. So if I'm B blood group, I have B blood group antigens that are being expressed on my cells, and I have naturally occurring antibodies against blood group A. Like that, do we all have antibodies against HLA that we do not express? No. Human beings do not, there are no naturally occurring anti-HLA antibodies. So I put that statement in red, and there is an asterisk here. Why is there an asterisk? I'll come back to that in a couple of slides. So for now, let's assume that there are no naturally occurring anti-HLA antibodies. So you need sensitizing events to develop anti-HLA antibodies. The common sensitizing events are prior blood transfusion, pregnancy, and a prior organ transplantation. Okay, now, how do we detect anti-HLA antibodies? We currently, the technology used to detect anti-HLA antibodies is a solid phase assay. Solid phase assay are reactions where one component of the assay, either the antigen or antibody, is sort of coated, or it is caught in a solid surface like a test tube or a bead, and the other component, either the antigen or antibody, is free to remain in the fluid phase and act. So a platform that is commonly used for this solid phase assay is the one that's developed by Luminex Corporation. It works on the principles of flow cytometry, and it is conventionally called as a Luminex test. This is basically a bead-based multiplexed immunoassay that combines proprietary microsphere technology with fluidics, optics, and digital signal processing. So what is this microsphere technology? You will see on the right this cartoon. Imagine there are 100 different beads. Each bead is coated with 2 different colors. And by shining 2 different lasers, you are able to identify what bead you're dealing with. So in a way, this is simply a multiplexed assay where in a small amount of sample, you are able to detect multiple antigens. Now, remember that this technology that developed by Luminex Corporation, this platform used, what is known as Luminex test, has nothing to do with HLA. It is simply a flow cytometry. It's simply a test that is based on the principles of flow cytometry, where you have beads, 100 beads, each bead is being identified as a particular bead. Each bead is given an identity number. So in other words, by using laser signaling, you'll be able to say that, okay, I'm getting light signals from bead number 21. I'm getting signal from bead number 26, so on and so forth. Now, in this technology, based on these beads, you can create whatever, or you can code whatever antigen you want on these beads. So example, if I want to look at cytokines, I can code antibodies against here, and then in each bead, and then use the blood sample or serum sample to get the information on cytokines. So how does this bead, or what is the output of this bead? So the single antigen bead output is reported as a mean or median fluorescence intensity value, or MFI value. This MFI value is a semi-quantitative assessment that does not reflect accurately the concentration or titer of the antibody, but gives beads relative fluorescence without reference to a standard. Now, this is very important to be clear that this is not in a linear scale. What does it mean? If I report that the MFI value for an antibody is 2000, and if I report for another patient that this is 4000, this is not equivalent to saying that there is twice the amount of antibodies that is present. So this is a relative standard, and this is not an absolute quantity. And above all, this is not a linear scale. Okay, so now we learned Luminex platform and the Luminex bead. Now, how do we detect these anti-HLA antibodies? Now, using this technology of this Luminex platform, 2 companies, one known as LabScreen and another one is known as LifeCode, they created assays to detect HLA. So how was this assay created? So if this is a Luminex bead, you generate a synthetic HLA on this Luminex bead, and this bead with this coated HLA is added, or serum samples are added to this. Now, let's say that this is A2 and my serum sample is added and I have an A2 antibody. My A2 antibody binds to this HLA-A2. Then a deduction antibody is added, and when the excess amount is washed, if there is antibody A2 binding to HLA-A2, then this deduction antibody that is bound to my HLA-A2 antibody is picked up as fluorescent signal. So this deduction antibody has a fluorescent labeled or a fluorescent conjugated antibody. This signal is picked up and reported as an MFI, mean or a median fluorescent intensity values. Now, routinely these days, what's used is a single antigen bead in which each Luminex bead is coated with one particular antigen. So if it is A2, it's one bead. If it is, say, B27, it's another bead. So each antigen is expressing only one HLA. Each bead is expressing only one HLA antigen. Now, where are these antigens coming from? The antigen source for the single antigen bead is recombinant cell lines. Now, when we use a recombinant cell line, there is a problem. What is the problem? Now, this cartoon is a structure of HLA class 1 molecule. You have alpha 1 or the A1, A2, A3, and then an beta 2 microglobulin chain. Plus, you have a peptide. So all these three, the HLA molecule, the beta 2 microglobulin, and the peptide together constitutes the antigen. Now, when it's derived from recombinant cell line and coated on the HLA beads, this is denature. So you don't have this entire structure going on beads, but you can have various combinations. Either you can have the alpha chain alone with the peptide dimer, or you can have an alpha chain and a beta 2 microglobulin dimer without a peptide, or you can have an alpha chain only. So in other words, though the single antigen beads are sensitive in the sense that you are able to detect antibodies against HLA of interest, we need to remember that this HLA molecule that's expressed on the single antigen beads do not resemble what is happening in cells because of the change in the configuration during the process of generating this antigen. Now, I said human beings do not have naturally occurring anti-HLA antibodies, and I put an asterisk there. So why is this asterisk? Now, single antigen bead assays, once they became popular, it was observed that there were presence of antibodies to HLA in humans, in healthy males, not immunized to HLA. So though we say that human beings should not have HLAs against naturally occurring HLA antibodies, once with the advent of single antigen beads, that such HLA antibodies were identified. Where are they coming from? These are probably cross-reactive epitopes in microorganisms, ingested proteins and allergens, but not against an intact HLA. So in vivo, they are not creating any problems, or they generally do not create any problems, because in the process of HLA molecule synthesis on a bead, because of the change in the architecture that I just described in the previous slide, you're exposing cryptic neoantigens. So far, so good. So you know that these cryptic neoantigens, there are these antibodies reacting. In vivo, these cryptic are hidden. So there is no way these antibodies are going to react to the antigens. So it's not a big deal. But this can be a problem, because though in vivo, such antigens are not normally accessible to these antibodies, when a test is reported as positive, to finalize the cross-match results or finalize the virtual cross-match result, that whether somebody has a real antigen or this is all because of exposure to these cryptic antigens, becomes a challenge. One way to overcome this problem is one protocol that's developed is this acid treatment of beads. When you treat the beads with acid, what happens is that further denatures HLA, creates more cryptic antigens, and there is increased MFI value of antibodies. So when you suspect some antibodies are not real, but are in the context of cryptic antigens or due to the presence of HLA antigens that have been altered during the process of bead generation, then one can use an acid treatment to see if the MFI value is increasing, in which case you are confident in telling that these are not antigens that are going to create problems in vivo. So far we dealt with single antigen bead antibodies detected in single antigen bead. Now, what is a donor-specific antibody? When the single antigen bead assay detects the presence of antibodies that are directed against donor HLA, then these are called donor-specific antibodies. Simple. Let's give an example. A patient who is being evaluated for a kidney transplant may have circulating antibodies detected by single antigen bead against A2, against multiple other antigen specificities. If this patient's prospective donor has HLA-A2 expressed on his or her kidney and every other cell, then this antibody against HLA-A2 is called a donor-specific antibody. Remember that though used generically, the term DSA, as detected in most laboratories, implies anti-HLA IgG antibodies. Let's go over this one more time. Though we call this as donor-specific antibody, for all practical purposes, donor-specific antibodies is anti-HLA IgG antibodies. What about IgM? They are not detected by the conventional Luminex instruments that most of the laboratories use. What about non-HLA? No, we can test for non-HLA, but the term DSA that's routinely used implies antibodies against HLA. What antibodies? Only IgG. Now, how is the single antigen bead assay reported? Now, this is a report from our own laboratory. Now, I'll draw your attention to a couple of things here. Now, if you see my pointer here, this is the individual self. Let's say it's me. I'm getting, this is my HLA profile. This is a deceased donor. This is deceased donor HLA profile. Now, what is reported here is DQ7. Now, let's go to so this DQ7 is the deceased donor's HLA phenotype. Forget the other antigens for a moment. So just focus on DQ7. I'm DQ2, my donor, my prospective donor is DQ7. Now, what is reported here is your single antigen bead assay. On the left is class 2 antibody results, DR, DQ, and on the right is class 1 antibody results, A, B, and C. Now, I have shown in red here DQ7s. So in the single antigen bead assay that's done, there are 5 different beads that are DQ7. That's what is shown here, DQ7, 1, 2, 3, 4, 5. So what is shown here is the MFI values. You will see that for these 3, for the first 2 DQs, there is one MFA value of 6325, another one is 6000. For the third DQ7 is 2500, and the fourth it's 925, and for the fifth it is 635. So they took my blood sample. I am the transplant, the prospective transplant recipient waiting for a kidney. They took my blood sample, put it in the Luminex machine, and then the output is reported as Muthu has antibodies against DQ7. But do I have an antibody against DQ7 that this patient, my prospective recipient, my prospective donor has? Because there are 5 DQ7s. So that's where you look for the next level, or if you see the field 2 of HLA. Now, there is no mandate yet in the US that HLA phenotyping has to be reported at the field 2 level. But most of the laboratories try to do that. Say for this patient, for example, donor high resolution HLA typing was available. And that donor's HLA typing was not just DQ7, that's your field 1, but at the next level, it was DQ319. Now, if you see here, go back, you will see here that this DQ7 of the 5 beats, one beat that is in bold here is DQB1 390. So against that HLA, there is 6018 MFI antibody. So that 6018 is reported here. Let's go over this one more time. I am the prospective, I am waiting for a kidney. My HLA is 2, DQ2. I get a donor and that donor's HLA is DQ7. But my HLA antibody is reported as DQ7 positive. So how do I know whether which subtype of DQ7 that the donor has or which subtype of DQ7 do I form antibodies? That's where this high resolution typing comes in. It turns out that the donor is this 0319 and the antibody against that I have in me is 6000 MFI. Let's say that instead of this 319, the donor has 301. So if you look at 301, it is only 635, which is below the cutoff. I will come back to that cutoff later. So which is below the cutoff. And then my report is denoted as negative, no antibodies against HLA DQP301. And then I go ahead with the transplant. So if you don't go to the second field level and then just stop here, I cannot be getting this kidney because I have antibodies against HLA D7. So that is where your field 2 level is helpful, where when you have an antibody that is detected by single antigen bead assay, you need to determine whether which subtype or which level or field 2 specificity that this antibody is directed against. Okay, so far I said there is a technology available. It's very sophisticated. You look antibodies, you know what the antigen profile of a donor is, and then you are matching or not matching and saying that this patient is ready to get this kidney or not ready to get this kidney. Seems so intuitive and straightforward. So what's the big deal? The big deal is that there are multiple challenges in defining the presence or absence of circulating antibodies against HLA. Absence of antibody is not equivalent to evidence for absence. What does it mean? A false negative DSA that's present but reported as negative in the single antigen bead assay. There can be several issues why there is false negatives. There is no beads for a particular specificity. I may be having antibodies against HLA-A2, but if the bead set that I'm using has no HLA-A2 that is coded on the bead, then this antibody is not picked up. So there is lot-to-lot variability in manufacturing. HLA structure differs between the vendors. So I said there are 2 vendors. And when I said the HLA structure is denatured during the process of bead preparation, there is a variability in the way the structures, the HLA structure is there on the bead between the 2 vendors. And then we discussed shared epitopes, where antibody targets that are shared by multiple antigens on the panel tested may result in dilution of the antibodies that bind to each antigen. So multiple beads have the same shared epitopes. My antibody that is in the serum is diluted out. So in vitro, it is reported as low level for each bead. Similarly, inhibitory factors that are present in the patient's serum, such as complement, C1q, may interfere with antibody detection. And this is known as prozone effect and require pre-treatment of the serum with heat, DTT, or EDTA, or dilution of the serum. So does this mean now, so we discussed about DSA negative, but DSA positive equals clinically significant antibody. Is that correct? Again, there is no easy answer for that. When to call an antibody as present or positive, or as absent or negative. Unlike the routine standard laboratory tests like serum creatinine or blood glucose, the way the HLA antibodies are assessed, those tests are not standardized. So at what MFI value to call a test as positive is not standardized, and different laboratories use different approaches. Most of them use a statistical approach, meaning that fluorescent level above a background. There can be a practical cut point as well, where you can simply try to correlate the MFI value with the physical cross-match result, or a clinical cut point where correlate with rejection or graft failure. Our laboratory at New York here uses 2000 MFI to call an antibody as positive or negative. But studies have shown that anywhere around 1000 to 1500 yield a high level of agreement among laboratories. If laboratories report virtual cross-match as positive whenever DSA is positive, then virtual cross-match is potentially more sensitive than physical cross-match, meaning that whenever a DSA is reported as positive, you call them as virtual cross-match positive, then you are picking up a group of individuals who are negative by CDC or by flow, but they are positive by virtual cross-match. So in other words, this boils down to the question that my DSA is positive, but my flow and CDC is negative. What do I do? So strategies used by laboratories to determine the specificity of antibodies is based on the sensitization history, knowledge of cross-reactive groups, epitope analysis, and refinement to the single antigen bead assay, such as serial dilution of the serum. So there is DSA positive prior to transplant. Now what? So when there is positive, so once there is DSA positive and say a physical cross-match is positive, flow or CDC is positive, depending on individual centers' risk-taking policy, how much they are comfortable with in dealing with immunological risk, one can decide not to transplant or try to do desensitization protocol before transplant or go in for a paired donor exchange. Now, if DSA is positive and the negative cross-match, this is what I said in the earlier slide, where I said if laboratories are reporting as positive DSA and the physical cross-match is going to be negative, then you will have a group of people where there will be DSA positive prior to transplant, a low level positivity, but cross-match is negative. So there, usually these patients do well and one may proceed with transplant. Now let's go to the opposite scenario. DSA is negative, but cross-match is positive. So you do a CDC positive, flow cytometry cross-match positive, DSA is completely negative. Now what? Now my chief here, Dr. Sudantran, he has an algorithm. We call it Sudantran algorithm, AAA. First here stands for defined donor allele. So first make sure that the donor HLA typing is done adequately at to a field 2 level so that the donor phenotype is clear. Next, A stands for check whether the donor allele is present in the single antigen B. In other words, I'm reporting DSA as negative because I simply don't have that particular antigen bead. So then, even though I may have antibody in my system or my circulation, my antigen is not, my bead is not simply picking up. Finally, check for autoantibodies, especially patients with lupus. They have plenty of autoantibodies. So all these, whenever you have a situation where DSA is negative but cross-match is positive, it's better to run an autoantibody where my own cells are allowed to react with my serum. So if my autoantibody values, say in a flow, if my autoantibody is positive and it is similar to alloantibody positivity, then it's probably all autoantibody that's causing this signal and one can go ahead. So in USA, laboratories must determine HLA phenotyping by DNA typing. However, they are not mandated to report it at field 2 resolution. But the problem comes because of the fact that single antigen bead assays identify antibodies against HLA at field 2 resolution. But because donor genotyping is mostly reported at field 1 resolution, this creates a challenge in interpretation of HLA compatibility. Let's go with an example. Donor HLA is done by DNA typing, but because it is not, there is no mandate to report the HLA at field 2 resolution, a lab reports it as B44. Now the recipient single antigen bead is at field 2 resolution. So it is reported as an antibody against B44O2 and B44O3. Now, without knowing whether the donor HLA is B44O2 or B44O3, it is not possible to determine whether this antibody that's present in the recipient is a DSA or not. Now, what about antigens that are other than HLA? We all know about MICA and anti-angiotensin 2 type 1 receptor antibodies also. So these antibodies that are non-HLA antibodies could be probably the prevalence of them could be more than what we think the prevalence is. In addition to allo antibodies, that HLA, MICA, and this ABO blood group antigen, we have several autoantibodies. Now, these days, they are still not commercially available, but several laboratories are trying autoantibody panels that are available using the same Luminex technology. How often to test for DNA after transplant? Now, this is controversial. Our center, in highly sensitized patients, we try to do it every 3 months after transplant, at least for the first 1 to 1.5 years, first 12 to 18 months. But how much or how frequent to test depends on the pre-sensitization, pre-transplant sensitization risk, as well as the cost involved. Now, finally, let's ask a question. So what if DSA develops after transplant? What is the big issue with that? The big issue with that is the slow deterioration of graft function and eventual graft failure. Now, transplant glomerulopathy is a condition that is due to chronic active antibody-mediated rejection. Patients who have donor-specific antibodies, whether it's a pre-transplant donor-specific antibody or a de novo antibody that's developed after transplant, over time, there is structural alteration in the kidney, which results in gradually increasing creatinine, glomerular and peritubular capillary endothelial cell basement membrane multilayering, with the result that there is proteinuria and eventual graft failure. This is our own study here at Cornell, where we studied 92 patients with transplant glomerulopathy or chronic antibody-mediated rejection. So I just want to draw your attention to 2 things here. One is that the patients who have this chronic active antibody-mediated rejection, their prior history of acute rejection episodes is only about 25%, meaning that the vast majority of chronic antibody-mediated rejection who lose their grafts over time, this is a subacute or a chronic problem that's going on, not a progression from acute AMR. So it's not that every person who has a donor-specific antibody develops an acute graft dysfunction. I do a biopsy, there is acute AMR, I try to treat it. The vast majority of them, there will be a slow deterioration in graft function. And again, so far, no proper treatment protocols are available, and and several groups are trying to come up with risk factors for identifying graft loss, as well as to develop effective treatment strategies. But as of now, how to treat, what are the protocol when there is a subacute or a chronic deterioration in graft function is not yet clear. So I will stop here. There are still several aspects of donor-specific antibodies and the consequence of donor-specific antibodies that need to discuss. But for want of time, let me stop here, and then I'll be glad to take any questions. Thank you.
Yeah, thank you, Dr. Muthu. I think that was a very lucid talk on a rather difficult topic, and I'm sure all the participants would have benefited a lot. It was very lucid. I just need 2 quick clarifications, if you can just take your name. One, of course, I understood that at night, when a deceased donor comes to your hospital, you're doing for the donor only the field 1. Am I right? You're not doing the field 2.
We are not doing field 2.
You're not doing field, you're doing field 1. That's fine. Now, my query is that you said that, again, I'll go back to what you said, that naturally occurring anti-HLA antibodies do not occur. So that would mean that in a male without previous transplant, without blood donations, you wouldn't ever have these preformed antibodies. But then they do have, and that's why we do test. So can you just clarify this dichotomy? I mean, from where are they getting these antibodies?
So these antibodies that were detected once the single antigen bead assays came into routine practice in, say, in a Caucasian male who has never had a blood transfusion or any issues before. So these are real antibodies. But what are these antigens that these are seeing? These are all cryptic antigens that are not going to happen within the circulation. But these antigens are being picked up once they are coated on the beads, and these antigens are being exposed during that process. But how did I, let's say I have this antibody, how did I form these antibodies in my circulation initially? That's what they say. These are all probably, the word probably is used there, probably cross-reactive episodes, cross-reactive epitopes against microbiota or against environmental proteins.
So they have no significance in that case. So then why test at all in a white male who has never had a transplant or blood transfusion?
Why test it all? Because to final, so in vitro, when you are checking them, they are no significance in vivo. But in order to know them, these are really cryptic. These are antibodies against cryptic antigens and not an in vivo pathogenic antigens. There may be problem in finalizing the cross-match result before allocating an organ.
Okay.
Let's say that I'm waiting for a transplant and I'm being screened. And if I have an MFI value of 3000 and tomorrow I get a kidney, somebody needs to determine that this is real antigen versus not a real antigen. So the cross-match results cannot be finalized unless beforehand I made sure, and as a recipient, somebody is writing in my chart that no, Muthu, in serial DSA analysis, he has antigen against A2, and this antigen that we are seeing is probably a cryptic antigen. Tomorrow, if I get an A2, go ahead. So that information should be available.
Okay. And one last question. So in your lab at night, are you going ahead only with a virtual cross match, or are you also doing a CDC before giving the recipient a graft? What percentage?
Madhu will be in a better position to answer that. I think their center, they close their eyes and don't even wait for cross-match, physical cross-match, and go ahead. Our lab types for their institute, so we know what is happening there. Not most of the centers, a significant proportion of our centers, including our own, when the DSA, when the single antigen bead assay is clean, we go ahead with the transplant. But legally, that issue is still not sorted out. By what is mandated, there needs to be the final cross match that has to be done at the center. Because this mandate was written in the time when virtual was not available, one can interpret that stating that this final cross match is a physical cross match. But because that's like a gray area, several centers are going ahead, but most of them are doing their physical cross match in the background. So in other words, we are not waiting for the results of the physical cross match, but going ahead based on the single antigen bead results. But once the transplant is done, then physical cross match result is also available. Madhu, can you clarify on that?
Yes, I think we have gone through two major transitions in the last couple of years. One is from in-person talks to virtual talks, and physical cross matches to virtual cross match. So you can see that the virtual talk works, as good or even better than the in-person talk at times. It's really nice and well done these days. And the virtual cross match, unfortunately, is not as well, deciphered. And the caveats, one of the first questions that came in the chat box is, the sensitivity, specificity, and pitfalls of Luminex assay. As you can see, there are many, and it's an evolving field, and that's why these, talks, which gives you the latest information as well as the lack of information, is very important. What we do is, when we have a patient who hasn't had any sensitizing events, and we have a sample at the immunogenetics lab within 2 months, and we look at the antigen, at the maximum level of, clarity that the donor center can give us, we look at the antibody panel and go ahead with the transplant if there are no major DSAs. And so that's what we will do. And if there is high PRA in the recipient, we would send a physical cross-match sample and wait for the cross-match we can always, watch more closely for an antibody-mediated rejection early on, post-transplant, and do what is necessary, and even consider an early biopsy if required. So, and also the virtual cross-match is sometimes, the coordinator on call at night is, tired, and they're going through all these antibody panels, working on multiple cases. So the virtual cross-match sometimes takes a little bit of time and headache to decipher from the reports. But thankfully, we have such an expert immunogenetic center with, Muthu and other members of his team running it. And so we have access to such knowledge, and, they have the answers at their fingertips when, we are at a loss. So that's a big advantage we have, which I know that many centers don't have. So a couple of more questions from in the chat box, which I want to, bring to Muthu's for clarification. One is, a question about how to detect eplets.
Okay, so eplet detection comes from high resolution typing, and there is a software known as HLA Matchmaker, so which uses computer algorithms to generate the eplet matching. So that was developed by somebody from University of Pittsburgh, and probably that's going to be the future, where you can go to a level of eplet matching. And already, as you know, there are papers that say that eplet matching is probably better.
I want to ask, relay a question by a wonderful beloved teacher, Dr. Gupta. He says, wonderful talk, made so simple. A patient has a CDC negative, but flow cross-match positive, but patient cannot afford single bead assay. What is the alternative in such a case to help the patient?
Okay, so patient cannot afford Luminex. Probably, again, some more details about the history is needed. Let's say this is a patient with lupus. First step is probably to do an auto. So to make sure that these are not all autoantibodies. And if there are no antibodies, if they are sure that this is an alloantibody, then probably the channel shift. How much is the flow positive? Remember that when we say flow positive, this is not a clear black and white. So what is flow positive? You have a signal, and then you allow the antigen, you have the antibody to combine it with the cell. When there is let's say again, let me go to my example. My cells are taken, my serum is taken and it is added. And let's say there is an antibody combining. So there is a shift in the fluorescence signal. This shift, you put a line at a particular level and say that this is your median channel fluorescence and your median channel shift. So several labs have different criteria for that. Say for our lab uses a channel shift of 40 to call it as positive. So then we need to determine whether it is 41 channel or 100 channels. Even 41 channel is positive. 100 channel is also positive. So it's like saying that if more than 120 is hypertension, 180 is also hypertension, 130 is also hypertension. So a sort of a clinical decision has to be made that, okay, this is only a slight channel shift. Maybe I will go ahead and see what happens when there is auto is negative. If it's definitely a 100 channel shift or a higher channel shift, then probably you cannot go ahead.
Another question from Dr. Narula is, a multiple blood transfused recipient, CDC negative, flow negative, TSA cumulative MFI about 6,000 to 10,000. Is desensitization required? The question is, whether you should go for individual MFI or a, cumulative MFI as a, significant.
Okay, so that again will be difficult to answer because the HLA, there is no one direct answer there. Because I told about cross-reactive epitopes, what will happen is, let's say that 3 different antigens are expressing the same cross-reactive groups, and I have one antibody, it can get diluted. So if the multiple epitopes are due to cross-reactive, then that cumulative MFI is more important. Let's say that I'm reacting to antigen 1, 2, and 3, and all those 1, 2, and 3 are shared epitopes. Then I'm having, one is 2,000, one is 3,000, another one is 4,000. Then against, these are all one epitope, but it is present in multiple antigens. But the cumulative MFI there is 9,000. That is different from saying that one antigen is completely different from another antigen is completely different from the third antigen. So it is difficult to answer in a straightforward way that if you go by cumulative or go by individual. It all depends on that cumulative is in a context of cross-reactive groups or these are all individual beats. So that's why close working with the tissue typing lab is important, and it is difficult. It's not like a standardized test like a serum creatinine where you just take whatever the lab says as a gold standard.
Many have asked this question in different forms. What is the cutoff of DSA that you'll use for induction therapy, and what kind of desensitization protocol you use? So you want to just allude to the, the use and misuse of MFI cutoffs.
Sure. So again, as I told, MFI is not a linear scale. So 1000 is not double of 500. It's a relative fluorescence, but it provides some information as a sort of a semi-quantitative assay. Now, again, as I said, studies have shown that about 1000 to 1500 can be used as a reasonable agreement among multiple laboratories. If you look at European papers, a French group have used 500. We use 2000. Why? Because in US, we use what is known as proficiency testing. ASHI, American Society of Histocompatibility and Immunogenetics, they send samples, standardized samples to multiple labs and ask them to report. And based on whether we are reporting it correctly or not, depending on how other centers are reporting, each lab is assessed. Now, based on that, we came up with a cutoff of 2000, and we have to change our your flow cytometry cutoff also to be on par with other labs to call positive or negative. Remember that this is not like there is no gold standard here. You are not looking at, say, I'm growing a bacteria in culture. That is gold standard. I'm doing a card test. Now I'm comparing it with that gold standard. No, there is no gold standard here. So what is the difference between 1500? 1500 is absent, 2000 is not absent. That is difficult to say. So as I said, most labs use anywhere between 1000 and 2000. Having done so many transplants where we also use CDC crossmatch and flow, approximately around 5000 to 6000 DSA, we get a flow positivity. And around about 9000 to 10,000 MFI, we get CDC. So in terms of sensitivity, you need at least like about 9,000 to 10,000 MFI value in a single antigen bead assay to have a CDC positivity, and about 4,000 to 6,000 in that range, around 5,000 to have a flow positivity. But again, as I said, this cannot be taken as for every antigen. This is like this cross-reactive groups. Several things come into play here. Now, what do we do for desensitization? Now, about 10 years before, desensitization was quite popular in the sense that there is no other option available. Now with donor swaps and you can put them in the chain where the swap is possible. The concept of desensitization is slowly going down. Madhu, maybe you can expand on that.
No, I agree with everything you have said. Any day, if you can, de facto decrease the immunological risk without desensitization, which is a much better option than desensitization. And there were a couple of questions on desensitization. There's another question interesting from Shamnad. If virtual cross-match is negative, how can we exclude non-HLA antibodies without CDC? I think even CDC or flow cannot exclude non-HLA antibodies, as far as I can tell. Dr. Muthukumar, you want to add to that?
Yeah. So in other words, if CDC is positive and DSA is negative, then obviously, because I said DSA is equivalent to, for all practical purposes, it is equivalent to IgG HLA, there could be a non-HLA, like your angiotensin 82 receptors or like anti-endothelial antibodies and other things. So there, at least, if you have that scenario and there is a real concern, most of the time we say, okay, this is some technical issue, your cross-match is fine, let's go ahead or something like that. But if cross-match is a real concern and the DSA is negative, then as I said, we use the 3A approach, making sure that the beads are, first we really did a proper HLA typing on the donor. The beads are really present. The antigen is present on the bead to make sure that I'm testing for what I'm supposed to test. So once we exclude all that, then there are options available that, though not commercially available completely, there are certain labs like Hopkins and UCSF that try to do non-HLA antibodies. There are both ELISA and Luminex techniques to do, platforms to do non-HLA antibodies. So again, so this is all coming in the context of that first transplant fails, you're going for a second transplant, the DSA is negative, cross-match is positive, what to do? But when everything is negative, you go ahead with the transplant, still you are, as Madhu said, you're not excluding a non-HLA antibody. You can only pick up retrospectively. Something happens bad, then you're saying that, okay, there is probably a non-HLA antibody.
So the couple of questions from Dr. Raja Ramanandran. One is a flow negative DSA positive transplant versus flow positive DSA negative transplant. Which one will be more which likely to have a more favorable outcome?
I'm sorry, repeat it again.
Yeah, whether you would count on a flow cross-match negativity or a virtual cross-match negativity as more significant, because flow negative DSA positive cross-match versus flow positive DSA negative cross-match, which one is more favorable?
Oh, I think in terms of, let me make this one more time clear. In terms of sensitivity, we are talking about DSA Luminex technology, which is more sensitive. It picks up antigen. Whether that antigen is really pathogenic, whether this antigen is clinically significant, we do not know. It is simply present. Flow is the next level where you're saying that I'm not only picking up antigen, but I'm finding antigens, finding antibodies that are binding to the antigen. Next, when you go to CDC, you're not only saying that I'm seeing antigen, I'm seeing antibodies, these antibodies are not only binding, but they are killing the cell. So obviously, in terms of sensitivity and specificity, CDC is a surrogate of what's going to happen in my body. If CDC is showing that the antibodies are killing the cells, then the same thing is going to happen in the body. If I go ahead with the transplant, the antibodies are going to destroy my kidney for all practical purposes. Flow means that there are antibodies that will go and bind to my kidney, which may or may not create problem. Luminex, if it is present, it will only say there is antibody present. It may or may not even go to the kidney. So obviously, a situation where your virtual cross-match, meaning that your DSA is positive by Luminex, but flow is negative, is better than a situation where flow is positive.
Good. There's a question on, I think it's pronase testing. Is it that, Raja?
I think it's prozone. Maybe you mean prozone, Dr. Raja?
Yeah. I just want.
Yeah, because that is probably a.
Yeah, pronase is a little more. I think we can talk about pronase separately. So prozone, what is prozone phenomenon? So in microbiology, we have all learned about prozone and postzone phenomenon. So what happens is when you have an immune complex, you need the right amount of antigen and antibody. When you have excess antibody, that's called a prozone phenomenon. And when you have an excess antigen, you have a postzone phenomenon. What is that? When you have an antigen antibody, when you do it an in vitro test, they bind and form a lattice. Its structure becomes big. So for that, you need the right combination to this chain to enlarge. If you have more antibodies, then this will not going to happen. And if you have more antigen, this is not going to happen. In a similar situation, happens in scenarios when you do a flow cytometry, when you do a Luminex testing. So what happens is you have an antigen, you have an antibody. Let's say you have an antibody that's binding there. An antibody binds to antigen. C1q, which is the starting point for your classical complement cascade, it's like a 6-headed monster. It has 6 heads. So those 6 heads is, in fact, why C1q is important to activate the antigen-antibody complex and move forward is because using its 6 heads, it is able to link multiple antibodies and sort of create a positive feedback to make the reaction happen. So in vitro, if you have C1q, this C1q, the 6-headed monster, can come and bind to the antibody. When it binds to the antibody, remember that in a Luminex, how am I checking what is this MFI value? I'm adding a fluorescent labeled antibody that goes and binds to the antibody that's present there, the anti-HLA antibody. But if the anti-HLA antibody site is already bound by C1q, my secondary antibody cannot go and bind. Let me explain one more time. I have a HLA antigen. I have a HLA antibody. So in a system, you have a bead where there is HLA antigen. My serum is added. My HLA antibody goes and binds. C1q that's present is now not allowing the fluorescent labeled antibody to go and bind to my antibody to create signals. That's why you will have a low level of antibody, low level of signal. So even though my antibody is binding, because my secondary antibody that's fluorescent label is not going and binding to this complex, there is no signal. So this is known as a hook effect or a prozone effect. So in order to overcome that, or there can be other inhibitory factors also, that is simply not allowing my secondary antibody to go and bind. To overcome that, you dilute the sample or do certain other things. One aspect is to dilute the sample. When you dilute that, you are diluting out the inhibitor that's present there. You're allowing the secondary antibody, which is fluorescent labeled, to go and bind, and then there is fluorescent signal. So this is known as a prozone effect. So what does this mean? So whenever a lab, this is all happening at the background in the lab. Whenever a lab reports or before reporting, they see that there is no antibody, they need to make sure that they did the right thing and there is no prozone effect. So one way of dealing with this, because calcium is important for this C1q to bind, one way is EDTA treatment, where you chelate this calcium. So our lab, for example, routinely does EDTA in all serum samples.
Fantastic.
Someone has suggested there should be separate beads for class 1 and class 2. That will decrease the cost. I suspect they may be talking about doing a separate test for the class 1 antibody versus class 2 antibody. Do you want to comment anything on that, Dr. Muthukumar?
I think Raja can comment on that, because single antigen, there is no issue with that.
So, Dr. Madhu, can you please repeat the question once, please?
So Dr. Manish Kumar Singla says, why can't companies make separate class 1 and class 2 kits? It will lower the test cost by 50%. So maybe it's given as a package, they will do the whole antibody instead of just class 1 or class 2 separately.
Indeed, I think the companies do market it as class 1 and class 2 separately. And we have the labs which only test for class 1 separately and class 2 separately, and also club them together. So it is available indeed. That's how it's reported also. Class 1 and class 2. You have the sequence.
Now, one way I can interpret that question is, instead of doing single antigen, let's rephrase the question. Instead of single antigen bead, now, because the vast majority of transplant patients are going to have negative, no antibodies, why waste money on single antigen bead? Can we simply do a class 1 as one test and class 2 on another test? If class 1 is positive, then go to single antigen bead. That's the approach that labs in US used maybe 10 years or 12 years before. But now, because the single antigen bead cost has come down, practically they have abandoned that, and it's gone to single antigen beads. Again, an analogy is that when COVID numbers were coming down, they started clubbing the samples. If 10 swabs are being done, you put all 10 in one PCR, and if that's all negative, you throw them out as negative. Then if one batch is positive, then pick up that 10 and do a separate PCR on each of that. So it's the same principle.
So the last question on the chat, hopefully I haven't missed anybody else, or if someone else asks a new question, we can pick it up. If one patient worked up for a second transplant as anti-HLA antibody present against his own HLA, it's again complicated that the previous donor was his mother, so there are some shared HLAs. Looks like autoantibodies. from Karthik.
Yeah, I mean, if it's going to be the mother's, I mean, these are going to be autoantibodies, so there is no problem there. The question comes in only when it is not a haplotype that's shared. Somebody else gave the kidney and my HLA DR4 and your HLA DR4 may not be similar. But if I get my HLA DR4 from my mother, that's all going to be the same because I'm inheriting that as a haplotype. But if I simply have a cadaver transplant or a disease donor transplant, that's DR4, there can be polymorphisms. And as I said, the field levels can be different. That's where your next field level comes in. If I'm having antibody against DR4, there are 2 DR4s available for me as donors, disease donors. My antibody is probably DR4 patient 1, not against DR4 donor 2. So that's where your field 2 level comes in.
Okay, another question has come up. Sorry. Difference between flow CPRA and single antigen bead CPRA. Let me also ask an added question to that. We had some patient who, sent for PRA and they said huge amount of background, they can't do a PRA. And what do you do in such a situation?
Huge amount of background in PRA.
Luminex PRA.
Yeah, I mean, like when background is an issue, I think the best option is to, I mean, again, that'll be probably be too technical to answer these questions. One, we need to make sure that your controls are fine. There are no issues with the beads. And if nothing works, then probably it's better to repeat the sample.
Is there an advantage in doing flow CPRA?
I'll come. Madhu, give me 1 minute. So there is something called as pan-DR reactivity. Sometimes what happens is, due to certain way the beads are arranged, you will have DR positivity for all DRs in the beads. So in certain situations when somebody has 100% CRA or CPRA, meaning that you have antibodies practically against everything, we need to make sure that this is not a pan-DR reactive. There are certain situations where you will have antibodies against every bead. That is your increased background. Versus a person with 100% CPRA, there you can make certain distinguishing features. Say, for example, if I'm having antibodies against my own HLA, then these are all part of pan-PR reactivity, for example. So that you sort of throw it out, either go ahead or repeat another sample with another bead set. Whereas if I have antibodies against multiple specificities, but if I don't have against my own, then it means I'm dealing with a true antibody where I'm producing antibodies against multiple antigen specificities.
Dr. Muthu, I have just one question. What is the current status of C1q binding DSAs? Are you doing it routinely or is it only on studies? And your comments regarding that?
No, we are not doing C1q. Again, after that Alex Lupi's paper in New England Journal, there was a big push towards C1q. But C1q probably, one, there are some technical issues. Two, it is probably a surrogate for higher MFI values of DSA, and it may not be a truly pathogenic. So I don't think most of the labs are doing C1q.
Yeah, in fact, Alex Luby said his own center is not doing it.
Yeah, he himself is not doing it.
Request comment on Luminex lysate method of cross-match using donor lysate for cross-matching, used frequently in India and not sure about its standardization. How sensitive or specific is that?
Yeah, I think Raja should answer. I have this question comes up, but unfortunately, I'm not qualified to answer that question.
Oh, come on,
Raja.
You shouldn't be saying that. See, it's even in PGI, initially there were reports of the lysate methods being tested. So what actually happened is, I think it is not a standardized technique, one. And it seems that, many of these kits were being provided free of cost by the company. So that was a main pushover for, doing these tests. In PGA, at least we have given up doing a lysate test, and we no more do this lysate, and we don't interpret it clinically, at least.
Yeah, I think Raja is absolutely right, and that's what it should be. Also, as he said, it was given freely, so I think many were using it, but I don't think it should be used. Yeah, fully agree with Raja. I think we need to conclude now, I think, because it's been a long time, and So if there
It's
are other
100.
pressing questions, so we can send email to Dr. Muthu, and I think he'll be happy to answer them. It was an excellent talk. I think we all learned a lot. He made so many complicated things so easy, and I think.
Thank you. Thank you.
Thank you, Dr.
Thank you. Thank
Thank
you.
you, Madhu, for being around and really moderating this.
Thank
My
talk.
pleasure.
Thank you.
My pleasure.
Thank
you
you.
very
Thank you, everybody.
Thank you.
Thank you, everybody. Thank you, Bomik sir, and thank you, Madhu. And
Real pleasure. Yeah.
so I cannot thank you enough. Thank you so
Thank
much.
you.
It was the first, invited series talk, and I think we have had 100 minutes. You have spoken 100 minutes. I think only a politician can match you. Thank you. Thank you.
Thank you. Thank you.
Thank you. Bye. Bye. Bye.
Bye.
Bye.
Bye.
Bye.
Rohit, just stop the recording.